All right, welcome today back to the Birmingham Medical News Ask the Doctor podcast. I'm Steve Spencer, the founder of the Birmingham Medical News. We are a news, as the name implies, we're a medical newspaper in Birmingham, Alabama. We write for healthcare professionals, doctors and healthcare administrators, executives, the whole range. We've been doing it now for 20 years. And this podcast today, we do this in partnership with Kassouf & Company. Kassouf & Company is a full-service accounting firm headquartered in Birmingham that have been in business for almost 100 years. They've got a full range of accounting, from all your basic accounting services for personal and business, as well as a wealth management arm. And for our purposes, they've got a healthcare practice that has been very, very successful in this area. So today, this podcast today is sponsored by HudsonAlpha. HudsonAlpha is a nonprofit genomic research institute dedicated to genomic research. One of the interesting things, too, is that when HudsonAlpha started, they brought in Richard Myers, who had been one of the original researchers when they first decoded the human genome. And y'all tell me if I'm wrong, but I believe he came from Stanford. Is that correct? Okay, yeah.
That's right.
So anyway, HudsonAlpha, they're using genomic research, studying DNA to try to work on everything from finding treatments for diseases like cancer and dementia to developing crops that are more resistant to pests and climate change. They have focused on health and, as I said, research for all sorts of things in North Alabama, and they also have somewhat of an educational outreach team as well. Anyway, today we're going to be talking about pediatric genomics, and we have two researchers from HudsonAlpha, Susan Hiatt and Meagan Cochran. And both of y'all, before we start, would you tell us a little bit about yourselves, your background, educational background, any work you did before coming to HudsonAlpha, and what motivated you to come to HudsonAlpha? Anybody
Sure.
start?
Yeah, I'll go ahead and start.
That's all
Hi,
right.
everybody. So, yeah, thanks for the introduction a little bit. I'm
Sure.
Susan Hiatt. So I started into science and into biology at a small undergraduate college in Pennsylvania.
Okay.
And then I actually went and got a PhD at Purdue University.
Okay.
And I did some research in genomics, but more in model organisms. So I did research in C. elegans. So it's a tiny little worm that people use as a model system, which is a lot of fun and can teach us a lot about genetics. And after my PhD, I actually went to NIH to RefSeq, which is a database where they curate sequences of genes and proteins and sort of help curate the human genome, to provide sort of a standard for other scientists who are doing genomics research. And I really enjoyed that job, but I really missed publishing, doing research, doing independent research. And so when I heard about HudsonAlpha, I was like, that's a perfect fit. And so that's when I really got into human genomics here. And so I've been at HudsonAlpha for about 10 years now, doing pediatric genomic research.
And Meagan, what's your background? Okay.
So I actually grew up in Birmingham, and growing up, I had this idea that maybe I wanted to be in healthcare, but I didn't necessarily want to be a physician. So throughout my undergraduate degree at Auburn, I bounced around from some different ideas. Everything from maybe being a pharmacist to maybe being a veterinarian to maybe being a high school biology teacher. And somewhere along the way, my career advisor said, "Hey, have you heard about this thing called genetic counseling?" And so when I looked into it, I found that genetic counseling really sits at the intersection of cutting edge science and research as well as patient care. And so that seems like a really good fit for me. So I did my training at UAB, actually worked at UAB for several years, and then came to HudsonAlpha in 2015. So I've been here about 9 years now.
Okay. Now, I think to start with, um, can you tell us a little bit? I understand there's a Smith Family Clinic for genomic. Um, um, is it for genomic health or research?
For genomic medicine.
Okay, I'm sorry. Can you tell us about that?
Yeah, absolutely. So the Smith Family Clinic for Genomic Medicine opened in 2015,
Hmm.
late 2015, with the idea to be kind of the first standalone genomics clinic in the nation. So
Oh, okay.
lots of places are doing genetics and have been for a really long time, doing
Uh-huh.
an incredible job. But the idea behind Smith Family Clinic was to be um, kind of the first standalone place with a real focus on genomics
Mm.
as this idea behind solving undiagnosed cases
Hmm.
of suspected genetic disease. And so we've been open now almost 9 years. Our 9 year anniversary is
Hmm.
actually just in a couple of days.
Oh,
And
wow. Okay.
so, and I've had the pleasure to lead the clinic for the past couple of years. And so over that time, we've really kind of expanded our reach and our patient base. So we see people all across the lifespan, from very tiny babies up through senior citizens,
Hmm.
all of whom have some suspicion of an underlying genetic disease that might explain some of the symptoms they've been having.
Um, what type of genetic diseases have you seen?
Oh gosh,
Hmm. A broad
everything. So,
range.
in the pediatric space, I would say
Hmm.
most of our patients are children with some sort of neurodevelopmental differences, things
Okay.
like developmental delay, autism, intellectual disability, children born with physical birth differences as well. And then as we go on through the lifespan, things might look a little bit different. We
Hmm.
see plenty of adults who were healthy until they weren't, and maybe they
Hmm.
developed some sort of adult onset neurological condition or
Hmm.
cardiology condition. But something about it has kind of triggered their healthcare team to say, I wonder if this could have a genetic cause. And that's how they get to us.
And so when the healthcare team has that question, when the patient comes to you, do y'all like sequence their genome or look to find?
Yeah, it really depends on the clinical scenario. So
Okay.
sometimes we meet somebody and it's a pretty obvious set of things going on with them that
Hmm.
really reminds us of one particular set of conditions. And so sometimes it'll be relatively simple genetic testing that we're
Okay.
ordering, maybe taking a closer look at their chromosomes or doing a set of genes that are all connected to one particular type of disorder. But
Hmm.
sometimes when we meet somebody, we say, well, I'm not really sure what this could be. This could be a lot of different things. And so in
Hmm.
that case, we might go wide and do something like genome sequencing or reading through
Okay.
all of the DNA to try to get to an answer.
Also, you mentioned autism. I didn't think, have they found a genetic component for autism?
Ooh, Susan, you want to take this one?
Uh,
I can.
because I didn't know that if
There
that's the
are
case.
there are lots of different rare neurological disorders where autism is a feature of that
Okay.
disease, and
Okay.
so there are a lot of um, so there are some specific diagnoses generally when there's autism along with other features.
Mm.
And there are definitely some autism risk genes as well, but it's a little bit harder to tease those out with genome sequencing if the only
Interesting.
phenotype is
Interesting.
autism.
So now, and I, this may not be something that you found, but maybe it's been found elsewhere. Like if you found several genomic sequences that put someone at risk for autism, does it, is there any data in terms of what percentage of that people with that risk become autistic versus who don't? I mean, is that how often does someone have the risk factor and it never happens, or would someone who have the risk factor for autism uh, never be defined as autistic, but may lean in some of those in that way a little bit?
Yeah, I think it's hard to say, and it kind of
Okay.
depends on what specific genetic factor we're looking at. So, like Susan said, sometimes it's autism plus lots
Okay.
of other features.
Okay.
And those we tend to think of as kind of these syndromic forms. Other times,
Mm.
though, there can be one or more genetic factors that says, like, well, this person is at an increased risk, but it may
Mm.
have been inherited from somebody in the family who has the same genetic difference,
Mm.
but doesn't have autism or doesn't have features, or maybe they have something different. Maybe they don't have autism, but maybe they had speech delay or something like that.
Mm. Mm.
Um, and so it's hard to predict, though, if we take a newborn baby and
Mm-hmm.
look at their genetics, what does life hold for them? That's much harder to predict,
Mm-hmm.
because most of the data we have is based on seeing people later on who are already affected. So we don't know how many people are walking
Mm-hmm.
around out there perfectly fine who have these genetic changes, but
Mm-hmm.
nobody's looking at them.
Yeah, because that's one of the things that's is interesting to me in that, you know, the epigenome obviously turns genes on and off, and we don't. I don't know if there's any data out there, not just autism, but any genetic um, um, uh, potential problems that never get turned on. And it's kind of, I would think, interesting to know why certain genes get turned on and don't. Like, any thoughts on that? I mean, could someone have a gene that's uh, makes them a little bit risk for cancer? Are there?
Go ahead. Do you want to go ahead, Meagan?
Yeah. So in general, yes, there
Okay.
are a lot of genetic conditions where if you have a variant or mutation in this gene, then you're going to
Mm-hmm.
have symptoms right from the get-go, right at birth, that are going to be really obvious.
Mm-hmm.
But there are a whole lot of other things where maybe there's 100% chance you're going to develop this condition, but not
Mm-hmm.
until you're 40 or 50 years old.
Mm-hmm.
Or maybe there's only a 60% chance. So you mentioned cancer. That's a really great example. There are a handful of genes that significantly increase a person's risk for developing certain types of cancer.
Mm-hmm.
But most of them are nowhere near 100%. Most of them
Mm-hmm.
can be anywhere from a 20% increased risk up to an 80% increased risk. And specifically in cancer, there's some theories for why that happens, but with
Mm-hmm.
other conditions, I think we're still working to understand why somebody can be healthy for a long time before symptoms ever start
Mm-hmm.
when that DNA difference was there for their whole life. Maybe Susan can comment a little bit on some of the mechanisms we think might be at work there.
Okay.
Yeah, I was just going to add a little bit about, like you were mentioning, the epigenetics or looking at all of these differences in the genome.
Mm-hmm.
So one of the things that we do, so I'm more on the research side compared to Smith Family Clinic, although we collaborate all the time looking at genomes of people who are still undiagnosed. And so on our side, over the last 10 years or so, we've sequenced 2,000 people who have some sort of suspected rare disease and looking for an answer. And in our studies and across the globe, across the United States and across the globe, most people don't get a diagnosis. There's only about 50% at best who get a diagnosis, even from genome sequencing. And so we know that there are some cases in there that surely have answers, but we just don't understand enough to find them yet.
Okay.
And so that's part of it. Like Meagan said, we're a lot of times sequencing people who we really think clearly have some sort of disease, something that is genetic. It has to be in their genome somewhere. And so some of the things that we're trying, there are definitely still a lot of genes that have not been described yet as disease genes,
Mm-hmm.
especially a lot of neurodevelopmental genes. We're still finding a lot of those, and we do a lot of research at HudsonAlpha in trying to collect enough patients to support publication of a new disease gene,
Mm-hmm.
and in some other labs here at HudsonAlpha trying to do some some of the experimental evidence that we need to support that something really does cause a disease. But the other things too, right, there could be something epigenetic that we just can't quite see in the basis of the genome, but how the genome is modified. And we're starting to look into some of those things. And there are definitely cases where there are changes in the epigenome that result in disease, that we're still just trying to figure out exactly how those work and which genes are related to those and that sort of thing. And there's also, even with genome sequencing, we think that it's the best, and it is definitely the most comprehensive test,
Mm-hmm.
but it still has some problems too. So not necessarily problems, but just limitations where there are some repetitive regions of the genome where it's just a whole bunch of one letter, a whole bunch of A's in a row,
Okay.
or GCC, something very repetitive. And so there's a few new technologies to do longer read sequencing that are really helping to identify some of these really complex odd looking changes in the genome that we just don't understand as well yet. But we're definitely getting there. We're starting to understand a little bit more of that.
What are some of the new technologies you mentioned? So you're saying that those can get kind of drill down a little further into the genome? Is that correct?
Yeah, so with a short read and long read, that's one of the big things we've been working on. And
Okay.
so the short read genomes, the way that most people sequence genomes, you have short little reads of sequence of letters of, a couple hundred bases, a couple hundred letters. But long read sequencing can be thousands or hundreds of thousands, even when you get into the super long sequencing. And so doing that longer read sequencing it seems to really increase sort of the diagnostic rate a little bit.
Mhm.
We've done a study. We actually just published our second study, talking about we have about 100 probands that we've published so far
Mhm.
who had a short read genome, and then we did a long read genome. And in about 10% of them, we find something that we couldn't see before. And so that
Wow.
technology of those longer reads really makes a difference.
Okay.
Um, and the other thing too with those long reads, they can also detect epigenetic changes. So they can detect methylation of bases,
Mhm.
which is generally a signature that shows that genes are turned on or off. And so
Mhm.
we're just really getting into that. Some other people in the field have already been doing this for a while too, and we know that that's probably going to really help.
And Susan, why? So over a person's life, the epigenome at times changes, right? At times, all of a sudden, it's not turning on a gene it used to turn on, or very. Why does that happen? Do we have a handle on that?
I don't have a good answer to that. There are lots of
Okay.
people here that probably do.
Okay. Okay.
But yeah, there are definitely different changes that just happen with aging and just over
Okay.
time, exposure to different things.
Uh,
There's changes in that methylation that are biologically relevant that just
mm,
happen because certain cell types need a certain gene turned on or off and things like that. So
mhm,
there is normal biological function to those changes in methylation,
mhm, mhm.
and then also trying to detect them too.
And I would assume
So
that,
it's different in all different tissues too.
yeah, I would assume that lifestyle is a big factor. Like I imagine if somebody spends their life smoking, there's probably not going to be really good changes in the epigenome. What about, do we know? Because I've always read that exercise makes such a difference in long-term health span. Do we know how exercise affects the epigenome?
I'm not the person to speak to
Oh,
that.
okay,
I don't
okay,
know for sure. Megan, do
okay.
you have anything
Yeah, I mean, I think
clinically related?
definitely. What I know about it and what I talk to my patients about is
Uh-huh.
it's less about affecting your DNA and what's turned on and off
Okay.
for exercise right now.
Mm-hmm.
But it's really more about you are born with some amount of genetic risk for any of these, what we call kind of our common complex disease. So
Okay.
thinking about things like diabetes, coronary artery disease, Alzheimer's disease, all of
Mm-hmm.
these different things. Each person is born with some level of genetic risk, and
Okay.
it's not due to any one particular gene. It may be a combination of things.
Mm-hmm.
So you can't really change that, but what you can do is change things like your lifestyle and behaviors and exposures and all of those things. And
Mm-hmm.
so if you imagine, a lot of times we'll talk about like the jar model. Like you have to fill up a jar in order to have the disease. So you're
Mm-hmm.
born with your jar part of the way full, thanks
Mm-hmm.
to your DNA. But can you add or take things out of your jar based on your lifestyle? So, if we think about something like diabetes, so things that would add to your risk jar would be things like having a sedentary lifestyle or having a poor diet or things like that.
Mm-hmm.
Versus things that would subtract from your jar would be things like good exercise, good dietary habits,
Mm-hmm.
not smoking, all of these different things that our doctors all tell us to do, right?
Mm-hmm.
And it's not so much about how they alter your DNA or the methylation, which there very well may be something to that. But I think right now the best evidence is for kind of saying, well, if all of these diseases are a little bit genetic and a little bit not, how can we influence the little bit not side to
Mm-hmm.
get ourselves on the right side of the risk equation?
And one thing you mentioned earlier, Susan, you talked about how somebody could have, if I understand it right, based on their genome, a 60% chance of, say, cancer or a 20% chance. That was really amazing to me. So you can actually look at their genome and 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 see what percentage that they're at risk. How does that work? Is it
Well, I was going to
that?
say, Megan, I think that was you, right?
Okay,
Talking
okay.
about
Because
the risks.
I never would have known that. I would have just assumed that, well, you have this gene that could give you, I don't know, prostate cancer. But you can
Mhm.
actually tell me, well, you have a 20% chance or a 50% chance. Is that, am I,
So for a while now, we've been really good at finding kind of high level genetic changes that
okay,
predispose somebody to risk. So
okay, okay.
people may be familiar with like the BRCA1 and 2 genes, the breast cancer genes.
Mm-hmm,
Angelina Jolie, there's the whole thing.
mm-hmm,
And
mm-hmm,
those are some of our real high value ones. So if you're a woman who has a harmful change in one of those genes, depending on where it is, your risk of breast cancer may be 60 or 80%. So we're
wow,
really
okay.
good at identifying those things and we
Uh-huh,
know the numbers associated with them. In more recent times, we're getting into the era of what we call polygenic risk scores. And those are where it gets really interesting.
okay.
Because when we talk about those high value, high risk things, we think maybe 2% of the population is going to carry one of those big risks. But everybody has some genetic risk for these various conditions. And so more and more we're seeing development of what we call these polygenic risk scores, where we take into account all of those small risk factors
Mm-hmm,
and can give somebody a personalized risk. And so if we say, well, the average risk for this disease is 20%. So 20% of people will
mm-hmm,
develop this condition, but you, based on your DNA, your risk is actually 30%, or your
mm-hmm,
risk is actually 5%. It's much higher or much lower than the average population.
mm-hmm.
What does that mean to you? And so that's something that's kind of been hanging out in the research world, but
Okay.
is slowly making its way into clinical care, is incorporating these polygenic risk scores to do things like decide, are you a person who your genetic risk is a little bit lower? And so if you do good diet and exercise, you can probably really swing your risk back.
Mm-hmm.
Or are you someone whose genetic risk is already pretty high? And so maybe I'm going to be more aggressive and start you on a medication a little bit sooner
Oh,
than I
like,
would somebody
okay,
else, right?
sure.
And so trying to kind of put people into a better kind of risk category
Mm-hmm.
is something that I'm really excited to see more of over the next few years.
It's really neat because I know you mentioned earlier the new technologies that can read more of the genome. So essentially, what you guys are telling me is it's becoming more and more, um, I guess intricate in what you can find, which is really neat. Um, One other thing before we move on to the second question, um, I know I know what CRISPR is, not a lot more than that. But is that something that y'all use at all?
There's a lot of research here that
Okay.
uses CRISPR in cells and in
Uh-huh.
cultured cells and things like that.
Okay.
It is a really useful tool so that we can, a lot of times, what we'll use it for
Mm-hmm.
is if we find a variant in someone and we think that it probably is the reason for their phenotype that we're seeing, the reason
Mm-hmm.
for their rare disease, we can put that variant into a cell and see what happens on
Mm-hmm.
a molecular level to see where that really fits in the pathways in the cell to see if there was more evidence that that is a variant that's really important and causing the mutation. So we will use it for that sort of thing
Okay.
um, pretty often. Yes.
Yeah,
That's
but
really
gene
neat.
editing in people is not something that is happening here at Hudson Alpha.
Yeah.
Now,
Sure.
there is research going on about gene editing in humans
Mm-hmm.
across the country and across the world because historically we've always said for the vast majority of genetic diseases, we can find it, but we can't cure it.
Right.
Maybe we can treat it, but we can't go in and change that actual DNA to get rid of the disease. But
Mm-hmm.
more and more now, people are saying, well, what if we could? What if
Mm-hmm.
we could go in and actually fix that DNA mistake and actually make a difference there? So again, research we're really excited about, but we're not actually doing that here at Hudson Alpha.
That's really incredible, and I've got to assume that at some point in the future they'll be able to do that. Um,
There are definitely some technological limitations in terms
oh.
of making sure that however you're delivering that CRISPR technology,
Mm-hmm.
that it's
Mm-hmm.
hitting the right tissue. So
Mm.
it's been really successful in diseases that are a disease of just the eye. Okay, we're going to do
Mm-hmm.
that gene therapy in just the eyes.
Mm-hmm.
Or
Mm-hmm.
it's a blood disease like sickle cell disorder. So we're going to go hit blood cells. For diseases that affect a lot of different parts of the body, it's more challenging to make sure the treatment gets delivered appropriately.
Okay.
And anytime you're messing around with somebody's DNA, you want to be really, really sure it's targeting the thing you want it to go fix and not messing around somewhere else.
It could be a horrible disaster. I can only imagine if you hit the right gene, but you have some overflow into other genes. That could be so obviously they've got to have it perfect before they could ever use it.
Exactly. So definitely research that it is happening and is coming, but for very good reasons is also super highly regulated.
Yeah, it has, right. The danger, yeah, I can imagine. And now this is a little bit, I'm moving on to a little bit different piece of what you're doing. Can you tell me a little bit about the Hero Fund?
Yeah, so that's something that we have available as a service here at the clinic. So it is essentially just a patient assistance fund. So we know that oftentimes families have a lot going on medically by the time they get to genetics. They may have been to lots of different doctors, had lots of studies and services. And so by the time they get to us, things might be tight. And so we don't ever want finances to be a barrier to somebody getting a genetic diagnosis. So the Hero Fund is offered to every patient who has an appointment at Smith Family Clinic. If they qualify based on income and family size, then the cost of their visit to Smith can be 100% covered. And if we end up doing testing like genome sequencing here at HudsonAlpha, the Hero Fund can actually cover the cost of that testing as well.
Okay. And I understand that you've also got a partnership with CRS?
Yep. So that's been going on since about 2020. We really ramped it up starting early in 2023. So this is a partnership with the Alabama Department of Rehabilitation Services, Children's Rehabilitation Service. And so CRS provides care coordination and clinics in all sorts of different specialties all around the state. But we are their genetics clinic now. And so if a child who is already in the CRS system, somebody thinks, hey, maybe this child could have a genetic condition. They refer them through CRS to our clinic. And once a month, CRS has dibs on the entire clinic day. So they bring their care coordinators, social workers, nurses here to our clinic. We see the patients. We recommend testing. CRS covers the cost of all of those things for those patients. And when we do find something, or even if we don't, but we say, hey, this patient needs this other specialist, they need this other thing, knowing that they're in the CRS network and kind of wrap their way around that is really incredible. And when we want to engage in research or say, hey, we thought we would find something for this patient, but we didn't. Hey, Susan and company, can y'all take a look? Kind of having that end-to-end care is really, really nice.
Well, that sounds great. I definitely appreciate you taking the time with us. Any closing thoughts? Anything you guys would like to add? Either one of you, it doesn't, and if you don't have to go to the stuff, that's fine too. You don't have to, but
Yeah, no, I think we're really excited about the work that's happening. As a clinician, I've been doing this for a while now, and every year we're finding more and more diagnoses for more and more people. And that's because the technology is better, our knowledge is better. And so getting more answers for more families has always been the goal. And I feel like with what's going on now, we're really getting to a place where that's really ramping up. So it's something that's really exciting to see.
Well, fantastic. Well, thank you all again for coming. And, I would just say I hope HudsonAlpha keeps up the good work. I'm sure you will. Thank you for listening to today's episode of the Ask the Doctor podcast, brought to you by the Birmingham Medical News and Kassouf & Company. Please make sure to like and subscribe, and make sure to tune in for our next installment. I'm Steve Spencer, and wish you and your loved ones a healthy journey until we see each other again.